Okay. Dr. Prahalad, hearty pranams all the my co-faculty members on the APDP program sponsored by AICT New Delhi, jointly organizing JNT University Anantapuram and JNTUA OTPRI Pharmaceutical Sciences. A warm welcome to today eminent personality and resource person and very good intellectual. More than the very good, the values and from the Harvard University, USA, Dr. KRISHNA Prahalad. You are very fortunate enough with such a intellectual at young age, and he's become doing the as a scientist with the PD and part of PDF program. With these words, I'm heartily welcome to the Dr. Krishna Prahalad for this FTP program. And one Veena, Pranati, is it clear my voice, Prahalad?
Yeah, I can hear, sir.
Okay.
I can hear. So you want me to start?
No, they're introducing
okay.
that one. 1 minute.
Oh, okay. Actually, I can introduce myself. That way, everybody feels comfortable, I guess. I can do that.
Okay.
If you allow me.
Okay.
So, namaste and hello to everyone. So, very good morning. So, myself, I'm Krishna Prahalad. So, first, I wanted to introduce myself as a student of Dr. Gopinath. Because such is the level of interaction we used to have during my bachelor's in pharmacy at Chalapati Institute of Pharmaceutical Sciences, Guntur. So the main reason for me today here is he told me like this is and I felt this is a very good program to interact with many of the personalities. So. So he told me to prepare introduction slides, speak about the journey of a research so that everybody gets at least some inputs. So I really like the idea of that because even I myself also have looked towards certain persons in my life who guided me throughout my life journey and who still are guiding me even now. So so my presentation will be divided into two types. So first I'll talk because as he mentioned, I'll walk you through the journey of how research, because I saw the topic you're having, it's like current pharmacy challenges, future and present and future, especially in the research part. So I'm going to focus my experiences, my observations, and my talking with other peoples and how my own self experiences in doing research, especially with more focus on pharmacology and toxicology. And then I'm going to present some of my examples, like what the work I have done till the point and what is the work I'm doing and my future interest and plans. So that if at least by the end of the session, if I could inspire at least one of them, then I would consider that as a mission successful. So, and I just want to make this session lively as possible. So if you guys, if anyone has any doubts or want to add anything, you can just raise hand in the Zoom, or you can stop and we can discuss it that way. So feel free, and I know this is a Saturday weekend, so but I made my presentation as lively to be as lively as possible. So try to relax and enjoy as much as possible. So without further much delay, so I'm just going
1 minute, Prahalad, 1 minute.
Sure.
Please be 1 minute. Wait. Veena. Veena, proceed and Veena, unmute yourself.
Yes, sir. Good morning, sir. A very good morning to all the respected stakeholders of 6 days AICT sponsored online faculty development program on pharmacy education and research for present and future challenges being organized by JNTUA OTPRI. I am K. Veena from the Department of Pharmacy Practice. Feels blessed to read out the vivid profile of an eminent personality of this morning session, that is Honorable Dr. KRISHNA PRAHALADH Sir Garu. Coming to educational qualifications of Sir, Sir has completed his B.Pharm graduation in pharmacy in Chalapati Institute of Pharmaceutical Sciences under Acharya Nagarjuna University in the year 2006 and 2010. Sir completed his master's degree in pharmacology and toxicology in National Institute of Pharmaceutical Education and Research, Naipur, Mohali in the year 2010 to 2012. Sir completed a Doctor of Philosophy in the Department of Pharmacology and Toxicology in National Institute of Pharmaceutical Education and Research, Sasnagar, Mohali. During his PhD, Sir worked on toxicological aspects of anti-cancer drugs and diabetes-induced testicular damages. During his studies with above drugs, he found that methotrexate causes zinc imbalances. Diabetes and hyperglycemia changes testicular and sperm zinc levels while passing through different maturation phases from testis towards epididymis. Coming to the accomplishments of Sir has received 7 honors respectively, and that is Rajini Bai V. Patel Parminova Award for Best Thesis in Pharmaceutical Sciences. Sir also received Sheldon M. Force Student Travel Endowment Award. Sir also received Graduate Student Travel Award. Sir has published 11 reputed national and international research articles. Sir has cleared 6 different national eligibility tests, and Sir has excellent merit record like 45th rank in GPAT, 856th rank in GATE, 72nd rank in CSIR NET JRF, and 150th rank All India rank in ICMR, and 45th rank in NIPER JEE, and All India 3rd rank in NIPER JEE PhD. And Sir has a very excellent experience. Sir worked in postdoctoral research in Brigham and Women's Hospital. He also worked as postdoctoral research Harvard Medical School. He also worked as postdoctoral research associate in the University of Rochester School of Medicine and Dentistry. And coming to, I am very much thankful to the organizing committee of this faculty development program for providing me this opportunity. Thank you, sir, and thank you, everyone.
Okay, carry on, Prahalad.
Okay.
Dr. Prahalad. Yes.
Thank you very much for the brief. I don't know what justification I'll make, but I'll try to engage as much as possible. So I know there are so many I mean, dignitaries here who are much more worthy to talk about their stories. But this is only about the opportunity that was given to me to talk about my story in the research. So I'll make that as simple as possible. And but stop me anytime, like if there are any doubts, questions, or any inputs anyone want to give, so that we can take it together. So without further delay, so as I mentioned, like my talk mainly covers two aspects. So first, I'll be discussing about the scientific journey which I took from the Bachelor's of Pharmacy to the till this point as a postdoctoral fellow here in Harvard Medical School. So and after that, my second part includes like some of the research aspects I took and how we proceeded and how we planned the experiments and how we carried out various aspects of toxicology and pharmacological testings. I guess everybody can see this, right? Can someone confirm? So I guess
Yes,
you
yes,
can see my
proceed.
okay.
Yes, sir, we can see, sir.
Thanks for confirming. So my topic of today discussion is on research in molecular pharmacology and toxicology. So currently I'm working at Brigham and Women's Hospital at Harvard Medical School, Boston. So first of all, a disclaimer. So there is no conflict of interest for the current presentation. It's totally my views and my journey. So the first scientific journey, like as I started, like I have did my bachelor's in pharmacy at Chalapathy Institute of Pharmaceutical Sciences, JNTUA. And there at the Chalapathy, I met Dr. Gopinath sir. So where he used to teach us all the pharmacognosy and natural products and in a very lively manner. So that's where I thought I also induced a kind of a feature where you need to engage any subject, given subject with the audience without getting bored. And he used to give us a live examples. And also I learned the very thing, the peculiar thing about his teaching and distinguished teaching, which I learned and I still use in my own presentations, is including a philosophical point, just not restricting to your scientific point. So I am a firm believer that all Indians are born philosophers. So everybody has their own philosophy, whether considered as a daily routine or considering as a presentation or considering as a research, etc. So I'm going to show like how it has helped me to nurture my research career and what I am right now. So and after stint of 4 years of a B.Pharmacy at Chalapathy, then I took we are the first batch to give the GPAT exam. I guess I'm sure the audience are here, teachers. I don't know. I'm not sure the broad audience, but I'm anyway, as I was indicated to talk about my scientific journey. So I'll be talking like what are the options available, etc. So it might be helpful for those audience. So then our batch is the first to take GPAT and last to take GATE in the pharmacy stream. So that has really helped. And the curricula of the bachelor's in pharmacy, especially with the university, has really helped us in preparing because we used to have sessions in the third year and fourth year for planning really to crack up exams. So everybody tried to inspire us to do a master's at least in the top institute. So that was our goal, at least. So we set a goal at least to study at Andhra University or something. But as you know, there is a saying like try for stars, at least you'll land on moon. So when we kept on trying, so I got like 45th All India 45th rank in the GPAT, which has turned that's a first turning point, like where after I joined, initially I joined and after that I took the exam for the NIPER and I also got 45th there. So then I took my master's in the pharmacology and toxicology stream at the NIPER Mohali, which is a prestigious, I guess, those who are working in the pharmacy stream knows about NIPER, which is solely dedicated as a research institute for the pharmaceutical education in India. So now it has expanded to the 7 institutes all over India. So if somebody is interested, they can encourage their students to pursue their research because every NIPER is developing their excellent institutes, and the number of seats are also increased as far as I know. And there is master's course and also PhD course, and also one can enroll for the postdoctoral fellowships at NIPER. So, and the NIPER Mohali is the first branch to be opened around in 1998, which is a national institute of importance. And there is an entrance exam for the bachelor's to master's levels, and also from master's to PhD level. So, and in the bachelor's to master's level, there is only single, first you need to qualify for the GPAT and then subsequently on the NIPER. And for the PhD level, you need to qualify the written exam, and there will be an interview and then deciding the place. And there are 13 branches, maybe extended to in the Mohali. And apart from that, there are recent pharmaceutical branches are also being because the pharmacy is growing as much. So they have recently introduced medical devices and industrial pharmacy too in some of the branches at, for example, like NIPER Ahmedabad. So they're doing research there. So the pharmacy branch is diverging as much as possible, because we are, after all, the leaders in the bulk drug productions and etc. So and after a stint of 2 years, I did my research project during my master's. I did on anti-cancer activities in the ion channels in the advanced cancers under the guidance of Dr. Tikku, who is the HOD and at the Pharmacology and Toxicology Department at NIPER. And after that, I shifted and I gave many exams, like for the PhD entrance at the institute, top level institutes, like I gave CSRNet, ICMR, and GATE again, and GPAT, and to qualify for the various exams. So I have attempted to go for the IIT Madras, CDRI, and all these things. But ultimately, finally, I decided to stay at NIPER Mohali. So due to the availability of resources and, for the research. So it took me like around 5.5 years to complete my PhD. So I did my PhD on the topic in the area of reproductive toxicology. And after finishing my PhD, then I have shifted for my first postdoc at the University of Rochester, where I have used my experience as a toxicologist, trained toxicologist, to further my research in the area of inhalation toxicology, especially with related to the smoking and also COPD models. And after 2 years of stint at the University of Rochester, I shifted at the last June to Brigham and Women's at Harvard Medical School last year. So currently, I'm at the renal division, working as mostly on the renal toxicology models. So you are going to see like how if you started with your career with one of the toxicology and basic backgrounds, so it helps to take your and shape your career. So first of all, as I said, like since this is a kind of journey, so everybody has their own story. I'll just briefly, I don't know what app it is, because Dr. Gopinath sir has asked me to cover as research, but I'll try to be as brief as possible. So first and most important, this is my first thanks to my family. So because these are really supportive and they have shaped my career to move forward. And like my mother and father, they told me to be different from my childhood. So being different is not is a good thing. So so try to be different and in thinking, and try to be too different. And one of the words which I like mostly is be fearless. So whatever you're trying to attempt is do it with your passion and be fearless, and don't think about and worry about what will happen. But it's only your passion which drives you forward. And my brother Praveen, he's also studied in the prominent institutes in the BTech cracking IEEE. So he worked at DICT, Dhirubhai Ambani Institute, and then at IIIT Hyderabad, where he's the person who inspired me to take up the higher education in institutes of national importance. So with this brief family background, so I move on to my further inspiration at the stop. So I'll tell you a story at the end of my slide. The story ends at the end of my slide. So it'll be a circle back story. So during my childhood, like my father used to say a lot about, I guess you know this personality, very famous personality, Dr. Yellapuragada Subbarao. So even in the biochemistry in the pharmacy second year, we used to have, I don't know like whether your curricula includes now, but nobody can surpass his experiments. So every pharmacist and pharmacy students need to go through his experiments. Such is the impact of his discoveries. So Dr. Yellapuragada Subbarao, and I'm happy he's from Andhra Pradesh to place and see this is only a tip of an iceberg, like what he has discovered. Like he is the first one to attain ATP as the energy source for the cell. And also he discovered this methotrexate and folic acid, which we now used for one of the essential list of essential medicines in the WHO medicine list. And his lab also synthesized the first tetracyclines, like chlorotetracyclines. And he has developed so many biochemistry methods which you commonly use. Even I remember today, we used to do during Chalapati, like second year medicinal chemistry labs, we used to do especially the lipids. So he has Fiske-Subbarao methods. So there are so many methods. So I'm sure like you guys have enjoyed his works. So he's one of the personalities that inspired me, kept on uh, to do research. And he also ended and I mean took up his career at Harvard Medical School. So everybody has, I feel that everybody has their own motivations in life. So these three are the real motivations of quotes in my life, which has kept me forward. And I would definitely say the power of quotation works in one's life. So so these three are really, the first quotation is the one which I used to, there is a big portrait of the Swami Vivekananda in the Chalapati library, which quotes the first quote. Even I see in the behind the Dr. Gopinath says, yeah. Because every day I go to the library, I used to very deeply. It's not like on the superficial. I used to experiment like whether this works or not, but trust me, it works. So finally, you'll enjoy, like if you are give a deep to this. By the end of your life, try to create your own. So you will be one day inspiration to one of the fellows. So much delay, and also with one of the kind of thing is he used to stories in the I don't know how he's doing right now. I'm sure like he is doing a similar kind of teachings, but that really is but I just want to share with everybody. So without much further delay, I'm going into the topic today, which has for the like I'll give you some of the insights, like my observations. Like a planning, if somebody is planning for a PhD on some of the ladder. So I'm going to particularly move on the especially in the toxicology field, because my background is in the pharmacology and toxicology. So the first lesson taught in the pharmacology and toxicology class is all things and all substances you see are poison. The only thing that harm, if you eat in a surplus uh, so the dose response, everything matters in all your studies. So these are basics taught, this quoted by Paracelsus, and some first risk. So where is the risk for the toxicity? So you should have a first your aim, like what you need to go and which direction you need to go. So here one of the slides from the Kesseret and Dowley, which is the classical textbook for the toxicology. Many used to follow. Says like toxicology may be divided into several types. So among them, like toxicology, it deals with the mechanisms, how the toxin acts and interacts with your body. And I'm sure like when the pharmacology deals with the pharmacodynamics and pharmacokinetics, the simple version they say is pharmacodynamics is like what the drug does to your body, and pharmacokinetics is what your body does to your to the drug. So it's a kind of the same thing for the toxicology, but here instead of the words, like you use the toxins, because the drugs and toxins are same, but if it is at the more dose, then it becomes a toxin. So, and this is a simple basic raw idea of that. And descriptive toxicology is also like a dose response, like how you plan your experiment and get the dose response and escalate from the preclinical animal models to the human ones, and how do you extrapolate them. And regulatory toxicology is one of the finest branches it's coming, because for any of the drug decision makings. So we have a specialized branch at NIPER, it's regulatory toxicology. That's the first branch started there. which has GLP certifications, like a good laboratory practices. So it's a very good training. Once, if somebody has a chance, you can visit NIPER Mohali, where you can see how it's planned, every experiment, and every experiment has a SOP. That's a very important in research. So everything you need to have an SOP, standard operating procedures, and how you do research. So you should write your every brief step to step in order to, because each in order to have a successful experiment, so you need to have a reproducibility. So for any of the research, first you need to have a hypothesis. So I like this. This is thinker stone in the back of them. So you need to you need to dive into your own thoughts and how to design your hypothesis. So that's the first step. One would start with questioning it. So I will explain. So my PhD topic mainly is on in the area of reproductive toxicology. So first, how do I identify a problem? So as I mentioned, so I took a risk. So I identified a risk. So what is the risk? So the problem is, so whenever the patients like are treated with anti-cancer agents, so there are several side effects. So it's the only thing is, like, you can extend extend the lifespan of the patient, but the quality of the life of the patient declines. So you need to also maintain the health. It's not all about how you extend it. So among the several kind of toxicities, especially I've chosen two of the classical drugs, like which causes the DNA damage. For example, like I have used cyclophosphamide and methotrexate. Again, the methotrexate, which is discovered by Ella Prakash Subbarao, which is still date used in that WHO classifications. And the other problem I have identified is with the diabetes. So these two are the major problems I have identified, especially with the toxicities. So there are several organ toxicities reported, because diabetes also have complications, so many complications there. If you get diabetes, like whether a type 1 is so much complicated, type 2 or type 3 gestational. So you need to get a regular checkups, even not only for your blood glucose, but periodical checkups you need to have for your kidney function test, liver function test, eyes, brain. So once you get the diabetes, I know, because my father is a diabetes, I know like how it will be like to have a diabetic, because you need to have restrictions in food, etc., etc. So such is the, and the other. So once first I identified a risk, then the question arises is, how can we manage the risk, or how can we? Is there methods to assess them? And finally, ultimately the question is, can I prevent it, or can I mitigate this using any of my knowledge? So that's where it comes the role of interventional pharmacology. So mostly I've used some of the interventional pharmacology, where based on the logical approach, we used targeting compounds. First, we identify the mechanism of toxicity, and then we try to use the logics to prevent it. So my model of thing is, because among the several of the argon toxicities associated, so I took the common problem is male argon toxicity, so male reproductive toxicity. So even the cancer patients who are treated, if they are, especially at the younger age, if they get cancer and treated, so their sperm quality gets decreased, and ultimately the children are affected. So it is the case with both male and female. So my part of research focus is on male gonadal toxicity or male reproductive toxicity. So, so to brief you, to give you the brief overview, like a simple pictorial graph. So I took my major target organ is testis. If you see that testis produces sperms, it's a cyclical production in the male. So after male hits the puberty. So once the sperm, which is immature when it's present in the testis, travels to the epididymis, and finally it gets maturation, and finally it gets ejaculated. So in the process of maturation, many of the organs help in getting the sperm maturation. For example, the prostate glands in man in the males helps in secreting prostatic secretions, which also helps in the sperm motility and maintaining the DNA integrity. If the DNA in the sperm gets damaged, then ultimately the children also affect. So now it's an established concept, like if the father or the mother who has a lifestyle changes, especially if they're habituated to the high fat intake or alcohol intake, or so many of the environmental pollutants they're exposed, their generation and their offsprings are getting affected. So this is true, and this hold with the case with both male and female. So this is the problem we have. So first, this occurs both in, so the area was chosen for my research, and then I proceeded, like the model system for I've used is anti-cancer agents and also the diabetes, because diabetes also affects the reproductive systems and the quality of reproductive and also the protections. So these are the, as I told you, during my PhD, these are my 3 philosophies I used to take, especially during my project work. So these, even my peers used to ask, why you keep these slides at the first your presentation? Then I say only one answer. So because my area is of reproductive toxicology. So I'm into the core philosophy of reproductive toxicology. So where I said like both males and females play a common role. Which was a concept. Earlier concept was only that thought that only females has a play a role in that. And also the last quotation says about the food habits. So really the food habits plays a very important role. Even a lot of nutrition and dietitian is taking a huge research in that area. So if you can see the animation here, so it shows how the sperm is travel from the testis and how it's getting maturated, and in the due process it acquires some of the proteins for its motility and maturation. So it travels from the different segments of testis, caput, epididymis, and finally it gets ejaculated as a final sperm maturation and gains the motility. So for this, it's a very tight regulated process. So if any of the process gets barriers, so it's or gets hurdles in the proper transportation, it severely affects the offsprings, and also it causes very other deformities. So next we need to plan on the objectives. So for that, we need to have a literature and a background about this. So in this, my objective is I found out like zinc is the one of the very crucial element. So even nowadays, if you see, zinc is one of the very crucial element required for one's growth. So even the WHO has mandated that the children of age, the neonates, especially those who are aged below 6 months, are required to give the zinc ORS. And now it is proven concept that if you supplemented the zinc ORS at the childhood time, so it will protect for your child till he reaches 3 to 6 years. So that is the very importance of the zinc element. So, and also it has got very multi-ubiquitous role. And luckily the other Indian who played in identifying this role of zinc is Anandas Prasad. He's in the Wayne State University in the US. So he's one of the very pioneer who identified this role of zinc. So now this has attracted me to take as a good target intervention agent, and I want to find out the role, like what is the zinc is playing, especially can it mitigate the toxicities, and can it improve the reproductive health of the male who is getting affected in the diabetes. So I want to target using the zinc with this reproductive efficiency. So for this, to mimic the human models, so as a preclinical pharmacologist, I need to have a model for this. So I have used rat models and using several of the interventional agents. So I have used rat. So, and I have 2 kind of models. One is to use anti-cancer drugs to induce the toxicity, and the other one is to induce diabetes in the animals. So first I need to have a model. So I need to check for the literature to have a suitable animal model. It may be for your research, you may select the first main important thing is, can you use the valid model for you? And if you are using that model, can it give you wealth of information, and can you do so many extrapolations? So that's what you need to first search. So according to literature, so I have used first and foremost important for any of the research is to have a literature search. So one should have a very good fingers in searching the literature and how to search a literature. So that itself is a very, very big topic to discuss. But these days, the online tools has become, and thanks to the 4G and 5G generations, I don't know, maybe the next generations also comes. So you can, thanks to the Google too. So there is a very good option in the Google Scholar. If you search for the Google Scholar, so you will get so many options, like how to search and make search, and you'll also get a patents, which will give a very vast amount of wealth informations on your research topic. And also I would advise everybody to make I'm sure like everybody has LinkedIn and all this, but I hope that everybody researchers also should have a research gate where you can ask so many questions with the peers around the world, and you can post, and you can also find simple solutions for your research sometimes, because it really literally connects the research world. So I would suggest everybody, like if they don't have, make a ResearchGate profile. So that's one of the very useful, where you can have a plenty of resourceful, and you can ask articles and resources. You can ask, that is a very good point of contact you can, where you can exchange with some of the scientists. You need not do in the work, but if you have some certain interest, you can directly message them in the ResearchGate telling so and so, I'm very much interested in this topic, so I want to know further about this topic. Are there any opportunities in this topic? And people, trust me, people are very much helpful. So, and they are willing to help. So if you are coming up with a genuine questions, so that's what's my experience too. So next, coming to the, once we have objectives, now I need to have properly plan and create a study design. So which is very important. And trust me, it takes a lot of pain to get a proper study design. So if you see here, I have treated the rats. So my model is completely usage of rats. So I have used the rat as a model because it, one of the things is it's widely used in the toxicology studies, especially with the male reproductive toxicity. So here I have used a 3 weeks model to induce the toxicity, especially to see the observed changes which I want. So as I mentioned in my first slide, the basic of the toxicology is the dose. So if you take the right dose, it will not affect anything. But if you want to create some of the doses where you want to observe the toxicity, so you need to have a strategy employing, where you need to do repetitive dosings, or you need to give a single dosings based on the clinical relevance. So here we have chosen the cyclophosphamide, which is a very well-known methylating anti-cancer drug, and used in several cancers, apart from methotrexate. So I'll show in the rest of the slides, I'll show methotrexate too. So here we have planned this thing, and we also planned what is the route of administration of this. So we have supplemented the zinc as the oral gavage, and we have given the cyclophosphamide as intravenous route. So I'm going to, and after once you design, you need to have a clear-cut idea, like what I'm going to look once I have planned research. So what are my different endpoints in my research to guide me and to give me a meaningful data. So if you observe this slide, so I have jotted down, I have taken these some of the very crucial and confirming tests to say whether my system is working and whether zinc has any role, especially in the anti-cancer drugs when we are giving it, or in the diabetes. So some of the endpoints which I have used in the study broadly classified in this. So trust me, the things you study in the B.Pharmacy or wherever in bachelor's, try to stick to the basics. So they help till wherever you go. So trust me, I have my bachelor's pharmacy books in my home till date. So so trust me, all this biochemistry and this will definitely help wherever you go. So try to, I mean, I don't know what the audience I'm speaking, but I assume that it will be helpful for everyone. So that's my intention of saying that. So I've estimated for the biochemical estimations, like various other oxidative stress parameters. And in order to observe, I have estimated various sperm parameters, like we have observed sperm motility in the microscopes. And by constant practice, you will be very much dexterous in getting very good slides, and you'll get so much experience by looking at the slides. You can say what's happening. I'm going to show you, and I guess you audience might appreciate, like by seeing the slide itself, you can say like what's happening in the organ itself. So these are the various endpoints I took for my study to get a conclusion. So for the finally, after you prepare a study, you need to have a solid results and discussions and how to write. So I have classified these slides because on the basis of how to write a paper and how to write a communications. So these are all the aspects it should run when you take up a research, and it should run at the background of your mind. So to show you some of the results, I'm not sure, because there will be a lot of results, but I'm just to the audience, like since this is a research about the research topic, I'm showing only some of the interesting observations we have observed during my research. So if you see, these are the sperm head morphologies from the rat. So the sperm was collected from the rat after the anti-cancer treatments. So here you can see the first one, second one, and third panels. So if you see the first one, these are normal. So those are not treated for the animals. But I guess you can clearly appreciate the difference in the sections 2 and 3. And also from the 2 and 3, so you should have a very keen observations while you are working under microscopes, and you should have constant patience. Patience is one of the very key factor while you take up a research. And the other thing is persistence. So you need to be constantly on the topic, and never give up, like if you are getting a result or not. So try again, try to think about, so why the system has worked for somebody, why it hasn't not worked for me. And it doesn't apply to the premier institutes. I mean, anywhere you can do your own research. The thing is, you need to have the lookup, because even during my bachelor's, I have learned so many things, and I used to question myself, and I used to put my small hypothesis, and I used to bother my teachers as long as, but they never discouraged me to by asking questions. So they tried to help me, and they gave me resource like where to find it. So that has really helped me during my PhD, and even it's helping me right now. So moving on, so you can see the other fluorescent stainings. So even we have used, this is a very simple stain. That's what I want to emphasize. So even in the common chemicals you have now in your pharmacy schools, you can do a very good piece of research and publishable in a good journals. Trust me. So, but you should have a very good hypothesis. So here I have used a simple stain. It is a very powerful stain. It's acridine orange, which is a DNA binding agent. So if you use, the specialty of this dye is, if it binds to the DNA, if you see the starting panel, first panel here, which is all green, the sperm heads are all green. So it binds to the DNA. If the DNA is not intact, it's not break, it will give one of the fluorescence, it will give a green fluorescence. But if the DNA got broken, so it gives a different fluorescence, where you can see in the second and the third panel, you can see they have orange colored stainings. So it means like DNA has got, the integrity was not maintained in the damage. So the second and third were from the anti-cancer treated rats. So here you can clearly appreciate that the DNA damage is clearly present by using this simple dye. So there is also other simple commonest dye, which is like toluidine blue, commonly used in the labs, which doesn't cost like around, it will be 200 rupees. And acridine orange is also very much cheap, but it is a very powerful microscopic technique, especially for the cells, if you want to observe. You can even observe in the compound microscope some of them. So in this way, you can do your own hypothesis. If you are working with not even the rats, even with the normal cells, you can do this, especially as a simple experiments, especially for the DNA damage integrity. So I used to work with the genotoxic assays. So it is a specialization of genotoxicity, is one of the specialization of the toxicology, where we assess the DNA damage response. So if you are any of the, even the environmental pollutant we breathe, so it produces a certain amount of damage response. So where our cell recycles the damaged DNA and removes constantly this process. But sometimes it doesn't get removed, so it is present there. So we need to detect those issues too. So we can use some of the simple techniques, but of course at times it is some complicated. You need to have really very good experimental hands to do that. For example, we have used comet assay, which are very sensitive in identifying a DNA damage, but I have not presented in this today's talk, but there are so many endpoints for that. So next, coming to the other powerful tool is histopathology. So if you see these are the histopathological slides from those treated animals. So I guess one would appreciate, if you see the top panel, the extreme end. I don't know whether you are seeing my pointer, but at the extreme end, where it is designated as CP above, it is the cyclophosphamide treated rats. So one can easily say, as I mentioned, like if you see the observation goes is, you can see there are less number of cells. The first observation comes into the mind. So next you need to pursue like what are the kinds of cells that is decreasing. So that's the reason you have like various quantifications. So in this example, I have used one of the steroids. Even in the medicinal chemistry, I guess I have learned this about the steroids, medicinal chemistry, and during my medicinal chemistry and bachelor's of pharmacy, where 3-beta hydroxy, 3-beta HSD, where the steroidal pathway and we used to draw these structures. So this is the enzyme allocated. The thing you are seeing on the histopathological sides, which is like a brown color precipitate, is the 3-beta HSD enzyme, which is a kind of steroid, which helps in the production of the sperms in the male systems. So you can see clearly the localization of this. So there are wide variety of techniques you can employ to prove a point, like whether what's really happening in the system. So these are kind of molecular techniques where you can use simple like protein estimations, what protein is changing and what steroid is changing, and you can estimate so many things. So, and moving on, and we also used immunofluorescence technique, like using microscopies, sensitive microscopies. So here you can see we have identified one of the protein which here you can see the red ones are colored here as is a propydium iodide. It's also one of the commonest dye. It's a fluorescent dye as a acridine orange. It's also like a cheap, but it's a very powerful binding agent, and it's commonly used as a DNA binding agents. So here you can counterstrain the nucleus, and if you see, we have used one of the protein called tesmin here, which is particularly present in the testis, male reproductive system at some certain stage of sperm maturation. So here you can see this. We have identified in our research paper that it settles between the nucleus and the cytoplasm during the maturation. So that's what we found out as an interesting observation. We have reported that. And moving on to the, I'm just, that's how you plan. So I'm just, I don't know how I'm good at my time, but moving on to the other slides, I'll quickly give it. So these are my other studies where I have used methotrexate as another anti-cancer drugs. So here you can see the power of histopathology. So you can clearly identify the clear structures. So histopathology is one of the very good technique, I guess one could employ and could do a very good research. So there are several societies, if somebody are interested, like histopathology, like veterinary histopathologists, like where they have societies, you can learn a lot of resources these days online, and there are separate protocols published each. And there are very simple protocols too, where one can employ in their own labs at your research institutes. So again, this is the epididymis we have isolated. Again, you can see various sperms. Even the previous slides, these are the sperms is present inside. Some are matured, some are not matured. So this is again chorda epididymis. This is really at the end of the epididymis of the sperm journey, where the sperms are stored. And moving on, if you can see, the methotrexate has caused various toxicities. Here you can see easily see the various different structures are affected. For example, in the second one, you can see the germ cells are coming out of the membrane, and it's going into the lumen of this. So this way you can identify several, and you can clearly see what are the different stages of sperms and different type of cells and how they are getting nurtured. You can even see Sertoli cells, which are nourishing cells for the sperms, and they keep the sperm alive till they grow and differentiate to the final ones. So my other one is like, I've also used diabetic models. So we have used streptozotocin induced, which is the commonest model. I have done with both type 1 diabetes and type 2 diabetes in animal models. So type 1, we have used streptozotocin, which is represented as STZ. So again, I have used the same zinc as an intervention method, as an intervention pharmacology. And I've done several molecular pharmacology, but I'm not going to present all the details and going to burden yours. So I'm just giving the interesting things. So the first, one of the commonest observations everybody thinks about diabetes is maintaining the blood glucose. So if you see this graph, so the zinc treatment, especially if you see, because zinc plays a very important and crucial role in the insulin secretion pathways and stability. Even one might have learned, and they might be teaching, like zinc insulin protamines, even still available in market. Even in the pharmaceutics, they study this. So if you see this, by intervention of the zinc, you can see like glucose levels are improved. And again, if you see, these are the pancreas, again, the power of histopathology and immunostaining. You can see here, the diabetes, almost all, it's not secreting any of the insulins here. But whereas the zinc treatment is showing the insulin levels in the pancreas. So when you got to work with one of the rats, so you need to learn multiple systems. It's not, don't confine yourselves to one of the target organ systems. So that's the very good, one of my PhD advisors used to tell me is try to look into the different, because you need to see a organism as a whole system rather than limiting to the toxicity. Of course, for the more information to deep dive, you need to focus on yourself on the one of the organs, as I did to limiting myself to the male reproductive organ. But you can see all the accessory organs where it is going to completely affect. So these are the changes observed in the pancreas. And subsequently, I also worked with the prostate and kidney and liver. So you can see histomorphology. So if you see the diabetes, which is represented as DIA, you can see what is the extent of damage it is causing. So, and zinc, sometimes it's helping in some of the organs, but no drug is perfect, because remember, we are trying to see the mechanism of this. And further histopathology, and you can see how the cells are getting damaged here. One would appreciate in the research, especially in the toxicology, and how they are coming back once you are treating the animal with this food interventions or any of the zinc, which is a very crucial trace element required after the iron. We have, because iron everybody knows, but after iron, the zinc is the next highest amount present in the body. Even if you come to the here in the US market, they have a separate zinc tablets. Even in India, I used to take the zinc tablets. So everywhere, nutraceuticals is also one of the targeting in the pharmaceutical industries, which is like very much high. So next one, as I told, I have also worked with the type 2 diabetic models. So where we have made the animals to eat the high fat diet. So we have used lard, so which is a fat from the pig. So and also we used to have gave a low dose streptozotocins to create the models. And again, you can see, you can appreciate the amount of cause. Even in type 2 diabetes, it's causing damage. You can easily see from the histopathologies and the scorings. And again, the same thing in the very enlarged versions. Like you can see the vacuole formations in especially in the second panels. And again, the observation goes like it's in the again in the epididymis. And we have done. You can see various cells how they are getting damaged. You can clearly see in the different groups. And this is also the kidney. So even the diabetes also affect kidney, and this is the liver. You can easily see in the diabetes there's the spaces increasing intercellular spaces, and it is affecting the architecture of the kidney. I mean, sorry, it's a liver. And we also done the immunostaining. You can see if you see the diabetes and the thing, it's highly decreasing this. We also give zinc deficient diet. So if you are feeding zinc deficiency, so you can see the last slide how it is. So almost all it's gone. The protein which is helpful is almost all gone. So such is the impact of the zinc which we found, especially what it's doing for the reproductive system, male reproductive system. So finally, you need to put a hypothesis if somebody asks you at the end of the day, so what your research talks about and what is it about. So this is the slide explaining what we have found out. So if you see the zinc, it plays a very important role even in insulins and to decrease the oxidative stress, etc. So we found out its association between zinc and other protein called GPX5 in maintaining the proper sperm maturation, and also in relations with the testosterone produced. So next, moving on, because I'll not take much time. I guess I'm good with time, I guess. So moving on next, so this is my PhD background I talked with you. So now one might wonder, so how it has helped me to launch my first postdoc position. So as you might see, I have my background in the toxicology. So then later on, I applied for my first postdoc. I met my mentor for my postdoc during a meeting. So this is how you make the first important thing. If you are planning to do research in future further, the first thing is to increase your circle interactions. So try to be interactive with as much as people as possible. The very good platform is to send a LinkedIn. And when I say LinkedIn, most of the people don't like just regular request. So you need to introduce yourself in a request saying that I'm so and so. I'm interested in this. So I just want to connect with you so that I want to learn and just interact with you. And at the first step, just don't ask like, I just want this opportunity, etc. First, make that connection. So even in the mentoring circles we take here, because we do some mentoring circles here with all young and budding researchers. So first thing they say is to use very good usage of LinkedIn. So it is worldwide, so it connects with everyone. So if somebody wants to take research and move on, so that's the first step to go ahead. So that's how I met my mentor during my research presentation when I came to the US, my first US visit trip sponsored by DST. So there I met him, and he's a very nice person to offer me a postdoc position in his lab. So my first postdoc in Rochester is involved with dealing with the smoke, because smoke inhalation toxicology. So smoke is the model where it induces the COPD. So I know it's very hard for some people once to get this COPD, the smoke. It is one of the very damaging and nasty things it's doing to our environment, and it's causing so many toxicology in the lungs. So my plan, as I mentioned, like what you need a careful plan once you want to do a research. So here we have used mice. So we have 2 mice which are having inherent fluorescent reporters in the mice. So they are tagged with the mitochondria. So, and we have exposed these mice to the smoke, and then we have extracted mitochondria, we have extracted lungs, we have extracted different cells, and we have done confocal imaging and tried to estimate various parameters like mitochondrial function test, which you can see. And we also treated with the exosomes. Exosomes are kind of a nanoparticles which is produced by the body. So initially these were considered as a dustbins that cells produce, but now the field of exosomes is growing like anything. So now it was very well established, and there are so many clinical trials going on, and there are so many companies in around Boston. Like I mean, at least I know like one company working where they want to develop this exosomes as a therapeutics. So these are naturally produced by the body, and especially the stem cells area is growing on. So stem cell produce so many of the exosomes, and even stem cells are also used for the treatment. So to get the results, so these are one of the confocal images where we took from the reporter mice we have. And these are the various estimations where we are done for the mitochondrial function test using seahorse equipment. So I'm not going to enter discussion, because I just want to give a glimpse how the research we have planned and how direction we want digging in the further molecular studies. And further, we have done these days, big data is a kind of word everybody is listening. So we further dig with so many genes. These are the data from the human patients. So we worked with the smokers, water pipe smokers, e-cig smokers, e-cigs are also, e-cigarettes are also coming. So we got samples, like serum samples, urine samples from the patients in the Rochester. So then we identified what are the genes it's getting affected. So we have done sequencing. So these are all, we have identified some of the RNAs, unique RNAs in those patients. So yeah, that's how we plan the research. And finally, coming to the end, like I'm almost at the end of my 1 hour presentation. So so right now so this is my first postdoc. So now again, I'm using the same basic toxicology principle and my basics, which I learned from the bachelor's, which is following me to apply in this position at right now at the Brigham's and Harvard Medical School. So right now here I'm doing my work on genome editing. So and right now I'm involved in current because this year the Nobel Prize was awarded with the CRISPR-Cas9 technology. Well, so we are working on this technology right now to do a genome editings, because that's one of the very hot areas where people are trying to cure the disease. So I'm not presenting much on that because we are still in the stages of producing our studies, but I'm happy to discuss at any time, like if you have any doubts on that. So again, that brings back me to the closing of the circle from the starting slide. So that's how I landed up at the Harvard Medical School. So where one of my childhood heroes, Dr. Subbarao, has done his research. So I'm right now at the same place, and I hope one day, I don't know, maybe I don't know how I inspired, or at least if I inspired one or somebody, it'll be that's a good thing in my life. So these are the first slide. He's my my PI at NIPER. The first is Dr. Gopinath. He at the starting day of itself, he says like PhD is not something research or something. It's relation you develop with the masters, and it also about philosophy. He also used to discuss about life philosophy and to take the things easily, so that then you have stable mind to do a research. And he is my mentor in the master's, HOD, and the Department of Pharmacology. And I have met during this journey, I have met so many mentors, peers, and nice friends. And I also kept my my interest. I used to do mime and skits. I used to write some of the dramas. So that's last slide is about that. I'm at the extreme end. So keep your passion for anything which you do. That's what I'm trying to say. And this is the first slide is my lab at Rochester. So he's my PI, Dr. Irfan Rahman, very nice guy. And these are my team at the Rochester, which I done my first postdoc. And to the extreme end, my current PI is Dr. Venkat Sabbisetti. So he did his he's also from Bachelor's of Pharmacy from Andhra University from 2000. So he's my current PI here at Harvard. So we are, so again, see the circling back. So pharmacy guys are working together right now here. That's what we are doing it right now. And some of the photos, I got a wonderful opportunity during my PhD. So I was selected by one of the young scientists to go and participate in Germany to attend the 68th Lindau Nobel Laureate meeting. So in that, every year they select on the theme. So the theme which we have went is physiology and medicine. So where we got a time to spend with 40 Nobel laureates for 5 days. So he's the Nobel laureate for identifying the circadian clock. And he's the one, Farid Murad, who identified nitric oxide, nitroglycerin tablets. So he's the one. So it's very nice to see those who identified those mechanisms, which is saving so many lives today. So he's the guy who got Nobel Prize in 1998, and they are still active doing the research. And finally, we had a privilege to have a lunch with the 2004 Nobel laureate, with the Aran Chichinova, who identified the ubiquitin system, which many drugs use till date. So as a pharmacy and pharmacologist, and the inspiring, you can see his videos in the YouTube where he started his career. He took his interest was as a medical doctor, but he came to the research career at 32. He got attracted. So he says, like, there is never a limit for the age to start your thing. So stop thinking like the age as a limit factor for yourself. If you have a passion, so you can do that. So that's the ultimate thing which I have learned. So, and with this, I thank you, and I hope this is a weekend for you guys. So I don't know what I have talked. I hope at least I have conveyed at least something about the research take, my research take. So I'm very much happy, and thanks to Dr. Gopinath sir for giving me such a good opportunity to interact with you guys. And I'm very much happy to answer questions, or if I'm not late with the time, I guess I'm stick to the time. So up to you now to interact if you have any questions.
Thank you, Doctor. Thank you very much, Dr. Prahalad.
It's my pleasure.
Very nice presentation.
Yeah.
See, no, you are the now time is almost 1:30, I think so.
That's okay. It's
I'm
1
more
o'clock.
active.
I mean, I'm always
1
active
more
to
o'clock
discuss.
after 80.
No, that's what
of the work.
No, actually, the important point I want to say is my first research was with you, because I remember, like, you used to do at the night beside our hostel room, you used to measure your activities and used to call us. You really used to engage us. So I really kept that passion with me. And now I remember, like, using a scale with the photographs, you used to bring a professional photographer to take all your slides during your PhD. So those have really impact on me, because you used to stay very late in the labs during your PhD. I saw how you did your PhD and with this thing. So you dedicated your time. So I'm really happy that you called on us. And I mean, even in future, if I'm of any of you use for you, I'm ready. I'm happy to do. So, I mean, if anybody has suggestions, I don't think like doubts, because I don't want to bombard with too much scientific information, because I know when you meant to say the journey, so how to take it. So if anybody has suggestions or any clarifications needed, so they are welcome to ask. If not now, they can mail us. So I guess if you type my name, I mean, it's publicly available profile, so you can see and ask. But if at all, if somebody has, I mean, they can proceed, or you can type maybe. And thanks for those nice words in the chat. And even I enjoyed giving a talk.
And my dear colleagues, you have any queries, please? And please be interact.
Somebody has raised a hand. He's from Dr. Jagadish. I see here.
Okay, Dr. Jagadish is please unmute yourself.
Dr. Krishna, am I audible?
Yeah,
Yes.
I can hear you.
So excellent session. So your the way of methodology and interpretation and the flow and the presentation was too good. And the last word, the survey bhavantu sukhinah. So this is what the need of our. So in this regard, I would like to have a small suggestion for your future studies. I seen a lot of data in this particular field, but I would like to make from you, because you are in the similar manner we are working. What I mean to say, most of the times when you have studied the smoking effect on the health problems, most of the smokers, they say that even the smoking is not only the one. If you go to street, the pollutant, and if you go to kitchen, the smoke which is present in the kitchen is also having a similar effect. So don't blame me. Don't ask me to stop smoking. So what they will have a show the escapism. In that particular case, what I mean to say, these business people, they create this curiosity, making that smoking is not only the one, another pollutant and carcinogen is also responsible. Don't quit smoking. But this is going impetus to the smokers not to smoke, not to quit the smoking. At the same time, that addiction, it will be compelling to do that.
Yeah,
As you are already in this particular field, what I want to say, so you are continuing as a research, you take this task as a the pollutant which is present in the kitchen, the pollutant which is present on the normal common people, they are going on the street and they are inhaling. Similarly, the smoker, in the three ways, how the body is getting affected with the carcinogenic effect, or it may be inflammatory effect, or it may be that is a cough causing. Most of the coughing is the biggest one. So this is the one thing. Another one is the smokers. They say that I'm using a filtered cigarettes, and there is a particular brand which will not make me cough. So it is good so that I'm having not effect. Even somebody confess that. Even somebody is smoking, they are living up to 100 years without any problem. So smoking is not the reason. So because you are already in this particular field, I would like to have some extensive study, because it is you are relevant to something societal problems. So I think if you put some as a logic in your research methodology, this part also, and put the research community this point, if it is clarified, I feel it is good. I don't know to the what extent I have gone through some articles, they are highly contradictory.
No, actually, nice you have raised, because I know like what your concerns is about. But right now I moved out of that research area. But to be frank with you, because our lab also, like past in Rochester, is also involved in the second-hand smoking, third-hand smoking. It's not about the direct smoking. So we also did the research in second-hand smoking, third-hand smoking. And also there are collaborators who are working, especially in India, where there's a presentation, one guy from the Maharashtra, where he's working particularly on the COPD models, where it is affected from the household. Even in the villages, still now they use the same pipe to do the fire, doing that. So they are working on that too, and trying to educate some of them. So yeah, I mean, I definitely understand your concerns, because even I am also of the same, because ultimately you should have that, that is the quotation why ultimately you should do something which is beneficial as part of your research. It doesn't matter just publishing, that doesn't matter. Ultimately, something needs to be given to the society. Thanks for raising that.
One question will be there.
Yeah. Is there any other from Sheikh Afroz Begum? Is there any other factor which mainly affect on toxicity apart from dose? Yeah, I mean, as I said, like there are several factors. But the dose is the first principle is the dose, as I said, like by the quotations. The other thing is the timings, also the timing and the type of interactions, because, and also the maybe yesterday's speaker has talked about pharmacogenomics, I don't know, individualized. So individuals respond to different, and their nutritional status. I mean, there are a lot of things which plays a role, but dose is one of the very important, and which is more related to the pharmaceuticals. That's the reason when we say dose, because even nowadays people who take paracetamol, etc. So they're you know, they don't know, because you need to educate them sometimes, because it's very unhealthy to your liver. It is very serious side effect here in the US, because so many research groups are there who are working on just paracetamol with taking too much overdosages. So yeah, there is time, etc., etc. So there are so many factors. And next question is, what is the role zinc element in sperm maturation? So zinc has zinc is like, it's a common trace elements. So it's first of all, it maintains the DNA integrity in any of the cells, and the same goes with the sperm. And also, while it is traveling under the maturation conditions, it binds to the sperm and maintains the zinc integrity, and it will not allow the sperm to be motile. So once it is at the later secretions, so it just gets out and come, if I can show you, and even in the female reproductive system, when the sperm fuses with the ova, it releases the zinc. As earlier thought was calcium release, but now it's called, there's a new concept called zinc sparks emerging. So it is both important in male reproductive and female reproductive system. So for maintenance of those, what should I say, both sperm and ova health. Does this, I hope that that does answers your question.
And Dr. Prahalad,
yeah,
if you follow the lifestyle modification with traditional system of medicines, like there's in the food is a medicine. I think so. There no any disorders and diseases. Can you justify this one?
Yeah, I mean, as I
Because-
told you, everything is like a lifestyle disorder. I'm sorry. You're saying something.
And lifestyle modification with food,
Yeah.
nutrition, food, good habits.
Definitely there are, for example, I would say like we have used the diet models, because the Western diet, if you see here, even in the US, like it's like a fast food centers are so many fast food centers because they have a high fat content, cheese content. I mean, there's so many issues with that's the model I mean, even when I say model, it's closely mimicked with the human one. So humans are habituated of taking that food. But food definitely helps. For example, lifestyle also. When I say this, you can see the papers by Elizabeth H. Blackburn. She is a Nobel laureate who identified the telomeres for the aging. So if you maintain the telomeres for the healthy aging, so she has a paper with the other group where they say yoga, etc., are kind of maintaining the telomeres so that's the healthy lifespanning. So there are, I mean, even the people like Nobel laureates are working on that kind of concepts, like for the nutritions. So you cannot take, because it's our integral part, what we eat or what we. So in a nutshell, like that's what even we used to. I remember your diet in Chalpati. I used to see your diet. I'm sorry, but yeah, it's just giving me my memories back with you. Yeah, for the fruits especially, because they contain good resources. You used to eat always. When I see you only eat fruits. Yeah. But,
Because
so,
now the much research are going on and spending the amount for the cure of the disorders and diseases.
yeah.
My object and aim and object is that why can't you there then the base? What is the reason for the diseases? Why can't they're not identifying that ones? And why not they're not doing the research on this aspect?
I mean, yeah, everybody's trying to identify, like, because we are such a complex system. It's not a simple one. So we have so many involving elements within us, and each and everything is to be in a coordination. So if there is mistake in one of the gene and at least one of the nucleotide, we'll have so many things. And we are surrounded by such an environment. So, and some are so sensitive to that. Some are very resistant to that. So it's again individualized everything. So yeah, it will be very complex. Even I will be happy like one day to see the exact cause, but there are so many causes. That's what even everybody is getting funded for, because we have so many diseases, so many cancers, even the cancer subtypes, some aggressive, some non-aggressive. There are so many things. So, but ultimately at the end of the day, yeah, everybody's trying to work for that. But since it's a complex, to be honest, because it's a very, very complex system, so we cannot pinpoint to one. But of course, for some of the diseases, it's very easily identifiable, but for some, it's really complex. Yeah. So, anyone want to interact, or I'm happy to answer.
My co-faculty members. I'm given to you unmute option. If anybody have any the questions, please.
Yeah, I mean, I used to be the same way. And I mean, I don't know who are here, but I used to be in the same way. And the one thing which helped me is to ask questions as much as possible, because I would be the first in Chalpati or wherever I go. I don't know, people see me different when I ask questions, but trust me, it definitely helps you. So don't think like, because everybody is in the learning stage, because it's not like somebody is testing for knowledge or something, but we are all here to learn. But if you don't have right now, it's okay. It's just, I know, a simple talk. I know, I don't know how I delivered, but yeah, if anytime you want to comment on the work or comment on the research methodology and you want to know more, you can shoot me a mail. I'm more than happy to answer, but just include that you are so and so that I can remember like where you are coming from.
And one question from myself, Dr. Prahalad, and all the faculty members are present over here.
Okay.
And because you reach in a higher level, and what are the teachers have to give the guidance to the students? how can they motivate like you people? Because you are the team of the around 6 members. All these 6 members have a very good coordination and elevated higher level.
Yeah.
And no, even our teacher is not reaching the PDF levels.
I mean,
Can
to
you
answer, to
narrate?
answer, yeah. Go ahead. I mean, when you say the inspiration, because it's up to even for me, because we never know in life. The only philosophy I used to have is the thing which you hear at some part of your life, it's going to help you, because irrespective, it may be a smaller thing or something. It will definitely help you at some stage of your life. Because I mean, apart from that, even I used to be the same thing, because I used to attend all the seminars. But I know, because we cannot directly, because trust me, nobody gets all the concept the speaker is talking, because the only speaker knows what he talks. Because and ultimate, but the only thing that ultimately at the end of the day, the take home message is, are you ready to take it? And are you, because do you want to be in a position? That's what I wrote in my slide. Have your own quotations and get inspired, and by the end of your life, try to make at least one of your quotation. That's what, because if you keep it's just keeping that example, I mean, it would be, the people say too much positive, but it's doable. Because that's what even the background, the portrait behind you, that what he says. First, believe in yourself. There are two aspects that one should believe. So first is like, be fearless in approach you take. I mean, but be rational. I mean, be rationalistic and be fearless. And other thing is like, first have confidence in yourself. And the second utmost thing is, have confidence on your creator. These two things, whoever might be, it doesn't matter where you are. Like there is something even the PhDs or philosophers use things. So that's a very important. Have your own philosophy in your life, irrespective of what you are and what you are from. That doesn't matter. But have your own philosophy, because everyone has their own philosophy. But try to stick to it and just move on. So I mean, that's what my opinion. Because I know, because everybody has their own. I'm sure there must be so many wise people, like, but it's only today is my opportunity to talk, so I'm talking. But I'm sure everybody has their own story here, who is attending here. But maybe I'm the smallest one here, I know. So, but I tried my best to tell. But yeah, but I'm happy to even communicate with you people. Like if anytime you need me regarding any other methodologies, or what you want to know, at least some career options, or I'll try to do my best as possible, or bring some others for the sessions. So.
Okay, on behalf of the faculty members of the FTP, and somebody posed yesterday questions, that is, can you do the PDF in India in the pharmacy sector?
Oh, definitely you can.
What are the institutions offering?
So for the
what
PDF,
is the procedure for that?
So I would say, like, for the PDF, etc. If you are directly, you need to have some qualification exam, or you need to apply for something, because first requirement is your PhD. And in PhD, at least these days, papers have become important. So try to publish at least small papers. If you don't have, I know that sometimes it's difficult based on your institutes, sometimes to publish in impacts high, but I can understand. But try to be at least published in a good journals. At least try to publish it. So once you have this thing, so you can apply. There are so many things. For example, CSIR. So every year they have a call for the PDF, or it's research associate. So it may not be PDF, it's called research associate. But the good thing I would suggest for everyone who wants interested in doing that further research is, if they don't have a kind of publications or anything, and have at least the PhD degree. So if you want, first improve your publication profile. So the first step I would do is to join a lab at least for 1 year or 2 years and get some publications, and then move. So in order to join this lab, there will be several advertisements in the lab, apart from CSIR and ICMR research associate positions. So you directly apply to the people where they give advertisements posting as called as research fellows, or what should I say, research associates. So there is no problem even if you join as a lower position, for example, senior research fellow. So this, trust me, this kind of methodology has worked for one of my juniors. So he was also in a similar position, like having a less number of papers, but he has taken that pain an extra step. I would say extra step rather than pain, to do that, to go extra mile of doing again lower step. But he got very good publications. Now he's back as postdoc in US, and he's doing well. So and also, like, I would also remember, like, Dr. Gopinath sir, I used to say during my bachelor's, he used to say, like, try to, I mean, especially for the bachelors, I don't know if the bachelors are here or any other, but as we're all students, it applies everywhere. He used to say, oh, try to keep your hard work at this stage, you'll enjoy, you'll get fruits in the later stage or something. So, but the thing is, I thought, like, okay, if you do well perform, then I'll get in the M-Pharm. So I have tried very much, and I got a good rank in GPAT 45. And then I said, like, okay, I'm got a good, but then that didn't settle me, because once I am in the system, I thought, like, okay, he said, like, I'll be settled now, but no, it's a constant thing you need to put effort on. But ultimately, at the back of your mind, finally it becomes your habit, rather than you taking as a hard work, it becomes your habit, and you'll like it as a due course of time. But it'll be very much painful in the starting, even in the research careers. Trust me, everybody will go to the lows and ups and lows, ups and lows. So that's the reason I showed you my family pic there. So, I mean, talk to people. You can cope up, and you can do several, because everybody in their life has their own risks. I mean, every job, trust me, even you might be seeing, I'm here, oh, okay, this thing, but even everybody has their own, what should I say, their tensions, etc. But it ultimately up to you whether you are taking or not taking. That's the second quotation which helped me. If you are still internally, you can do so much externally. So try to keep still in self. So that's the quotation. If you see, that's my second quotation, which I have written in that one. And yeah, ultimately try to, yeah, stay positive. I mean, everybody says this, but it's very difficult to do practice, but definitely. I'll give you my mail ID. I mean, somebody are asking, I'm going to type in the chat. You can just mail me, but in the subject line, just include some tag like your student or your acquaintance of Dr. Gopinath or something, or some you met in the conference, I mean, this session, so that I can easily identify you. So yeah, I've just typed my mail ID there. So if somebody wants to have any comment or something, I'm most welcome. So, or criticism or anything. So I'm happy to take. But it's really, I'm happy because Gopinath sir, I mean, he's, yeah, we used to have such an interaction, lively interactions. I mean, I'm not exaggerating, but for other people, maybe they don't know our associations but that's what I'm saying. Is I get inspired and I'll inspire. That's what the thing ultimately at the end of the day.
And all the you people, even observing you from the even bachelor degree, even now postdoc,
Yeah.
same this one innocence in the face. That is the greater thing at maintaining. So when reaching the even the heap level, had to maintain the same innocence in the heart.
Yeah, definitely.
No, that is the greater thing.
Yeah. Definitely.
I believe strongly that the nature always support when doing the disseminating the knowledge or sharing the knowledge to the further generations.
Yeah. Definitely.
And almost now time is 2 o'clock and 18 hours of the work day.
No, that's right.
Thank you very much.
Yeah, thank you. Thanks, and it's I know it's a weekend, but thanks for your participations, and all the best for everyone who are here. And if at all I've inspired, like it's the session worthwhile. So and thanks for this wonderful opportunity for giving me, and hope to see everybody sometime.
Yes, another one, Prahalad, Dr. Ramakrishna is joining. If you want to be there in some more time, if you want to be, or otherwise we'll meet tomorrow.
Okay, I mean, his presentation, I mean, is right now?
Dr. Ramakrishna? Dr. Ramakrishna? Maybe some more time you will taking, I think, other.
I mean, I can stay. Yeah, he'll be also an excellent speaker. He's like a top 1% in the bachelor's. So we used to have a healthy competition, because healthy competition is always a good factor rather than keeping grudges, etc. That's what I observed is most of the faculties or somebody, you shouldn't go that side. So you should have very, I don't know, healthy competition. The competition should be there. But yeah, he was also very, I'm sure like he will disseminate some knowledge. I'm pretty much positive on that.
Okay, and there are no more questions, and thank you very much. And recently I'm narrating the one story.
Okay.
One of the child is becoming I think the higher cadre, SPR or DSP, but whereas his father is the constable.
Yeah, I've seen the story. Yeah, inspiring.
Here in Andhra Pradesh.
Yeah.
And when is the first she's the giant, she came to the office, and her father will be there in the near to the door, giving the salute to her. See, that is the professional way, is much elated.
Yeah, definitely.
You people, all the team of the very good team, maybe may God bless you, all the your team people, further heights. Maybe you'll get the Nobel laureates from the India.
I mean,
certainly.
As I said, like, I mean, ultimately, it's not about the prize. I mean, at least a thing we should do for the society. I mean, in this mortal body, that's the ultimate goal for everyone. Should I say? Because earlier I used to have the same thoughts. Oh, we should get this prize, that prize. But ultimately, as we progress, move on, that I realized, because that's what even I met with I went to the Nobel laureate meeting. There are like 45 Nobel laureates. We get to spend the time for 5 days. So they are so simple and humble. I mean, they're down to earth. You should believe that they come and talk to you like anything, and they say same thing. Like it's just their passion which has driven to them, and they even go to the lab till at the age of 80, 83. So that's really inspiring us to keep move on and compare our clocks.
Okay. Thank you very much.
He's joining. I can wait, like if he's joining.
No, I don't know. He's not. Maybe he'll join within 10 minutes. Ramakrishna and Veena, please give the vote of thanks to the Dr. Krishna Prahaladh.
I mean,
Veena, unmute yourself, Veena. The faculty member of JNTUA OTPRI.
Thank you, sir. Our sincere, heartfelt gratitude to Dr. Krishna Prahaladh sir, who has presented whole gamut of information on research in molecular pharmacology and toxicology. Thank you so much, sir, for inspiring us towards research methodology. Sir has clearly explained the various aspects of reproductive pharmacology like identification, management, prevention, and assessment. Sir has rejuvenated about some of the eminent personalities and power of quotations which has proposed by them, like Yellapregada Subbarao sir garu, Swami Vivekananda garu, Kanchi Paramacharya garu, Srinivasa Ramanujan garu. Sir has clearly explained about schematic representation and the importance of zinc in the process of reproduction. Sir has shared some of the experiences during SAR studies, and SAR evaluated various tests like biochemical estimation, sperm parameters, histopathology, and morphometric analysis. SAR has shown pictorial representation of rat sperm and morphology of head of rat sperm, and shown quasi-normal, glossy abnormal sperms heads respectively. SAR has clearly explained various objectives in study design. Thank you so much, sir, for sharing many memorable moments with us. Sir, we are very much impressed with your interpretation of results with pictorial representation. Thank you, sir, for our kind acceptance of our request to energize the registered participants with sound and knowledge on research in molecular pharmacology and toxicology. And thank you, our beloved Principal Gopinath, sir, for giving me this opportunity.
Thank you.
Thank you.
I'll take a leave from all of you. Thank you, and all the best for everyone.
Thank you once again. Prahaladh,